Hello everyone, welcome back to O-Level Oasis. Up till now, we've covered 4 chapters from your O-Level Biology 5090 syllabus. You'll find detailed lectures and past paper practice for each chapter in the dedicated playlists on my YouTube channel. And for some quick revision, don't forget to check out the quizzes in the YouTube Shorts section as well. Now, it's time to jump into a brand new chapter, so let's get started. Today we're starting with Chapter 5, Enzymes. We'll be going through this topic in detail, just as it's outlined in your O-Level Biology syllabus. This chapter's also split into 2 parts, and obviously, we'll kick things off with Unit 5.1, which is about enzyme action. In this part, we'll go over all the important stuff you need to know for your syllabus. According to specification number 1, you are required to describe a catalyst as a substance that speeds up the rate of a chemical reaction, but is not itself changed or used up during the reaction. But what does that actually mean? For example, think of it like, you're at a party where 2 people are too shy to start a conversation. Then someone steps in, introduces them, and gets the ball rolling. That person doesn't join the conversation, they just help it begin. That's what a catalyst does in a chemical reaction. A catalyst speeds up the reaction by making it easier for the reactants to interact. But here's the important part, it doesn't get used up or permanently changed. It does its job and comes out exactly the same. So, a catalyst isn't a reactant. It's more like a facilitator, it helps things happen faster without becoming part of the final result. That's why catalysts are so vital in both industrial processes and living systems. So, let's focus on the key definition you need to remember. A catalyst is a substance that speeds up the rate of a chemical reaction without being changed by the reaction itself. To put it simply, a catalyst makes the reaction happen more quickly, but it remains unchanged at the end. It doesn't get used up or become part of the final product. Let's quickly walk through these 2 diagrams to get a clear idea of how a catalyst works. Starting with the first diagram, this shows the reaction without a catalyst. You can see that the reactants need a high amount of energy to start the reaction. That's called the activation energy. The gap between the energy of the reactants and the products represents the overall energy change of the reaction. Now, if we look at the second diagram, with a catalyst, you'll see that the activation energy is much lower. That's because the catalyst offers an easier route for the reaction to occur. But here's what's important, the energy difference between reactants and products doesn't change. So, in simple terms, a catalyst speeds up the reaction by lowering the activation energy. It doesn't get used up, and it doesn't change how much energy is gained or lost, it just makes the process happen faster. Talking about the types of catalysts. There are 2 main kinds you need to know. First, we have biological catalysts, which are called enzymes. These are special proteins made by living cells to speed up reactions in the body. However, enzymes are quite sensitive if you heat them too much, for example by boiling, they stop working. That's because high temperatures damage their shape, and their shape is crucial to how they function. Then we have chemical catalysts, which include substances like acids or metals. Unlike enzymes, these are much more heat resistant. You can boil them, and they'll still continue to work because their structure isn't affected by heat in the same way. So in short, enzymes are powerful but delicate, while chemical catalysts are tougher and more heat resistant.